Center for the Ethiopian Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, students. This is Asrat. Our lesson today will be about photosynthesis. This is another very important energy transducing process taking place inside cells. Of course, animals cannot photosynthesize. They don't have the very crucial organelle that helps us to transform light energy into chemical energy. But plants can do that because they have this chloroplast that can harness the light energy and change it into food. The food that the whole world consumes every day. Every world consumes every single day is made by plants, algae, and some photosynthetic bacteria. Let's go to the details of our lesson. You know that this process is said to be photosynthesis. It just combines very simple inorganic molecules that are called carbon dioxide and water to produce glucose, the main respiratory substrate, which eventually is stored into starch and eaten as a food in our spaghettis, potato, and any other plant products and animal products that are transferred in the food chain, and finally produces the very important molecule called oxygen that we breathe to survive. So now, let's go to the detail of this process. How do plants harness that light energy and transform it into chemical energy? We used to define photosynthesis that way. It is the process by which plants convert light energy into chemical energy. Sometimes we define it as a process by which plants combine simple inorganic compounds like carbon dioxide and water to produce glucose, organic molecules, that means. Okay? So, this stage actually has several processes. I want you to know these very important lessons by the end of this topic. One of the lessons is locate where light-dependent stage and light-independent stage of photosynthesis take place, because it has two stages. The second one is you should be able to tell the products of light-dependent stage of photosynthesis and the light-independent stage of photosynthesis. We might not be able to see this in the laboratory in the class, but it should be done in the laboratory. But you know that chromatography helps us to separate pigments that are found in a chlorophyll. You know that chlorophyll has several pigments in it that can be easily separated using paper chromatography. And you also be able to explain photorespiration, why photorespiration happens in C4 plants, and how plants evolved from C4 pathway to C3 pathway, how photorespiration happens in C3 plants, and how plants evolved from C3 to C4 pathways and even CAM pathways. Lately, you will also be able to explain, differentiate, tell examples of C3, C4, and CAM plants. Follow me. So, photosynthesis. In photosynthesis, light energy is used in a series of reactions, especially in the light-dependent stage of photosynthesis. in order to make a light, very important organic molecules that are released in the process. And as it passes, it actually passes to, or is basically trapped by, the chlorophyll molecule that is found in the thylakoid membranes. And these thylakoid membranes are found in the chloroplast. So I want you to see that very important stages. I already mentioned these very important points. Let's go to those parts of photosynthesis. You can see that this is a leaf of a plant. Sorry, it is a bit darker, but you can easily see this is a leaf, and we took just this portion of a leaf, and when you magnify it, you see a very well-organized, layered part of a leaf. Just, it is layers. From upper surface, which is the upper cuticle, epidermal layer, protective layer of the leaf, and there is a layer of cells called palisade cells. We call them the mesophyll layer, together with the spongy layers. We have also several, but the main photosynthetic parts of the leaf are the palisade cell, or the palisade parenchyma, and the spongy parenchyma. These two together, we call them mesophyll layer, or we call it the photosynthetic tissue. They carry very large number of chloroplasts inside them. This is a chloroplast, the other energy transducing organelle in addition to mitochondria. This organelle, as you can see, has double membranes, the outer and the inner membrane. Inside of it, you can see that a stack of thylakoids, we call them the grana. You see this one? Each of them are the grana, and every single coin-like part is said to be the thylakoid. And it is within this thylakoid that the chlorophyll or the photosynthetic pigments are embedded. And these photosynthetic pigments are the ones that trap the photons of light and cause the very important processes of photosynthesis that are used in the latter stage. Okay? So I want you to know these very important parts. Chloroplast has double membrane. It has its own ribosome. It has its own enzymes. Everything is just well equipped in it. It is a self-replicating organelle, just like mitochondria. It has its own DNA, and it has its own RNA molecules, obviously. So they are able to replicate themselves independent of the mitochondrial layer. It has very important parts called two parts. One is the granum, as I have mentioned, which contains the stacks of thylakoids that contain the photosynthetic pigments. And it also has a liquid-filled section called the stroma. It is in these two very important parts the main stages of photosynthesis take place. Let's go to the stages. So, in that part of photosynthesis, we have also this very essential part called the photosystems. The photosystems are the biochemical pathways, okay, by which chlorophyll absorbs light. And inside this thylakoid, as I have mentioned earlier, there are very important photosystems that are called photosystem 1 and 2. And these photosystem 1 and 2 absorb different wavelengths of light in order to maximize the process of producing more energy that is required for the light independent stage, which makes obviously the food, glucose. Okay? So this photosystem 1 and 2 will help in the process. We will see the details in the coming lessons. Okay? You will understand more in the next session. So this is where actually the photosynthetic pigments are found. This is the structure of the photosystem. The photosystem consists of these very important parts. Okay? There are very important molecules called the reaction center molecule. This one, okay? It is called chlorophyll A. And there are, you see, there are many different photosynthetic pigments that can receive different wavelengths of light. And that transfers the energy until it passes to the reaction center chlorophyll, then to the electron transport chain as it is received by the primary electron acceptor. Okay? So this photosystem consists of very important parts that I want you to know: the pigment molecules, chlorophyll A, chlorophyll B, carotenoids. Sometimes you might find xanthophylls. Okay? The reaction center molecule, to which all the electrons that are initiated from different photosynthetic pigments arrive, called the reaction center molecule, chlorophyll A. And of course, the antenna complex. All that is called as the antenna complex. So, are you ready to go to the more detailed part? Let's go then. This is the chloroplast, as you can see. And this chloroplast undergoes two important stages of photosynthesis. Light-dependent stage. We usually traditionally call it light reaction. The light-independent stage, we also call it the dark reaction. The light reaction happens, obviously from the name, it requires light, and it takes in water and produces oxygen that you take right now. The oxygen that you breathe in is actually the result of the photolysis of water into hydrogen and oxygen. That's why we call water is life. It gives you oxygen when it splits. The hydrogen is actually taken to glucose, to carbon dioxide to make glucose, the food you eat. It is your life. Save water. Okay? And this process happens in the thylakoid membranes, especially in the grana generally. Okay? And this light reaction provides ATP and NADPH. NADPH is like NADH in the cellular respiration. Okay? It just added phosphate and hydrogen. This hydrogen taken from the water is provided to the next stage. So this is light independent and this is light dependent stage. Look what light dependent stage gives to the light dependent stage. ATP, NAD. ATP, how? Made through photophosphorylation. You see, photosynthesis is a self-sufficient process. It provides the energy it needs for itself from itself. No need from other sources like mitochondria. It is a self-sufficient process. And the light independent takes place in the stroma. Okay, so those are the very important processes that I need you to see in detail. How does it happen? Light dependent stage. The light dependent stage of photosynthesis involves two photosystems. Photosystem 2, Photosystem 1. By the way, it starts with Photosystem 2. It's not to confuse you, it's just how it is discovered. Scientists discovered Photosystem 1 first, then they have discovered the other photosystem next. Then they called the first one here called Photosystem 1 and Photosystem 2. What happens in the light dependent stage? 1. Light energy is required. 2. Water splits into hydrogen and oxygen. 3. Photophosphorylation happens. What does that mean? Photo, light, phosphorylation, making ATP or addition of phosphate. Plants make ATPs that is required in the light independent stage from light. So that is ATP. They follow the same kind of electron transport chain, but it is 2 here to make the ATPs. Okay? So, let's see how it happens. There is a photosystem 2 here. I want you to identify this because photosystem 2 and photosystem 1 are frequently asked questions on your national exam. Photosystem 2 absorbs a light wavelength which is around 680, while the wavelengths of light absorbed in photosystem 1 is 700. You know, the lambda is a unit for the wavelengths. And as long as the light energy is absorbed Okay, here in the photosystem too, what something happens is the electrons in the chlorophyll on the photosynthetic pigments in the antenna complex of the photosystem start to get excited when light starts to strike them. These electrons start to get energy from light and start to be displaced from the chlorophyll pigments to the reaction center molecule, which I said the chlorophyll A. And this electron starts to jump to the primary electron acceptor. And this primary electron acceptor passes the electron to the second, called plastocutting, passes it to the cytochrome complexes, passes it to plastocyanins. These are, by the way, the protein molecules that help in the electron transport. Okay, I told you. The light reaction of photosynthesis involves 2 electron transport chains. And it uses those molecules to pass from one electron to the other to the other. In the process, ATP molecules are made. In the process, again, this electron now comes to here, and another electron from this photosystem 1 again gets displaced, excited, and goes to the other primary acceptor from the reaction center molecule. It goes to ferridoxine, NADP reductase, until it makes all the ATPs required. All the ATPs are made here. All the NADP molecules required to be reduced. One photosynthesis or one glucose takes around 6 water molecules, which means 12 hydrogen molecules will be combined with NADP and taken to the light independent stage. I just want you to know some of the electron transport chain. Distinguish these two for your national exam. Okay? So the light dependent stage happens in this way. To see more in detail, you don't need to get confused with these pictures, but just to see more in detail if you want to, this is the thylakoid membrane. The thylakoid membrane taken from just here, okay? This is the thylakoid, you see? This is the grana, of course. Just take one piece of it and just see a very small section of that. If you just slice it and see the detail, it has photosystem 2 and photosystem 1. And look how the hydrogen ions are pumped. The photons of light are coming here, strike the electron, electrons get excited, and they pass through different electron carrier molecules to the photosystem 1. Photosystem 1 again passes it until the whole ATP molecule is pumped and produced. Hydrogen is pumped and ATP is molecule produced. And this hydrogen molecule finally goes to NADP. And this NADP now finally will be able to make a reduction process in the light independent stage to make glucose. So that is the whole process. Electrons in the chlorophyll molecule get excited. They pass through different photosynthetic pigments. They go to the reaction center molecule, which is always chlorophyll A, primary electron acceptor, electron transport chains, photosystem 1, then it goes to the other electron transport chain until it finally produces the ATP required for the next stage. By the way, the next stage is called the Calvin cycle. And as well, ATP, NADPH, pass to there. So this is the whole stage that we have discussed so far. The summary of the process. Photons, water splits. Don't forget it. We call it photolysis. Water splits into hydrogen and oxygen. All right? Second, ATP is made. You see? How many ATPs do you think are made? 18 ATPs are made for phosphorylation. Reduced NADP. Reduced means it carries hydrogen here, okay? Separately. Where does this take place? This takes place in the grana. In the light-dependent stage, remember all those things. The antenna complex, the photosystem 1 and 2, photolysis, the electron transport chain, and the grana and the thylakoid. They all combine. Of course, ATP synthase, the one that produces ATP through chemiosmosis. I'm not going in detail of this because electron transport chain is the same in case of cellular respiration. But sometimes plants could suffer from cyclic photophosphorylation. The whole process that we dealt with so far is non-cyclic. You have seen that the electron that is excited from photosystem to goes from one pigment to the other, then primary electron acceptor, then the electron transport chain, then it goes and it goes until it is received by NAD. P, don't forget P, NAD pH, then goes to light independent stage. But sometimes, due to some problems, like, for example, this only happens when sugar cannot be synthesized for some reasons, such as lack of carbon dioxide. Due to lack of carbon dioxide, sometimes this process could be cyclic, so that the electron that is excited could recycle. It goes to ferridoxine, plus to quinine, cytochrome complexes, plus to, you can see that it returns back to chlorophyll while it should go to NADP. It happens in such cyclic way, only in photosystem 1. We call it cyclic phosphorylation. Okay, this reduces photosynthesis. So you should be able to identify non-cyclic and cyclic photophosphorylation. ATP is made, of course, but no glucose is made during cyclic phosphorylation. How is carbohydrate then made? What is made from light-dependent stage, students? ATP, NAD pH, and water splitted. Oxygen is gone. It's coming to you now as plants make food during the photosynthesis process. How is carbohydrate made is the next stage of photosynthesis. We call it the light-independent stage. The light-independent stage sometimes is called as Calvin cycle because it is named after the scientist called Calvin. Melvin Calvin, actually. It's a funny name. So, it involves 3 important stages. Carbon fixation. This is a process that actually fixes carbon dioxide from the atmosphere through the stomata and combines it with a molecule waiting inside called ribulose bisphosphate, just like oxaloacetate, okay, in mitochondria. Reduction process which involves NADP, okay, and regeneration. Let's see how each of them happen in the process. Okay, for this process, of course, we need some very important enzyme called Rubisco. Rubisco is an abbreviation of the most abundant enzyme on this earth called ribulose bisphosphate carboxylase oxygenase. This enzyme is a very important enzyme in the process of photosynthesis because it helps us to fix the carbon dioxide, combine it with RUBP, which we call ribulose bisphosphate, and start the cyclic process that eventually makes the food. Food is good, you know that. So, let's see each process as it happens. We have mentioned plants taking carbon dioxide from the atmosphere through their stomata, use light from the sun, and very important wavelengths of light, you know those ones. Especially the blue and red light spectrums are the most important light spectrums used during photosynthesis. Of course, most of the light is absorbed. Green is reflected. You know that's why leaf appears green. But the 2 light spectrums are very essential. So what happens in the Calvin cycle? Carbon dioxide. You see, 3 carbon dioxides are absorbed once for one or for a single Calvin cycle. These 3 carbon dioxide molecules, as you can see, will be combined with a 5 carbon molecule called ribulose bisphosphate. 3 ribulose bisphosphates combined with 3 carbon dioxide, 1 for each, that means. Then you can see that as it is not stable when it combines with carbon dioxide, it breaks down into 6 3-carbon molecules called glycerate phosphate. And these 6 glycerate phosphate molecules now use ATP, okay? The ATP made during light dependent stage. They use ATP, and this GP becomes reduced from GP to TP, glycerate phosphate to triose phosphate. This is what I said, reduction process, the second stage. Then this again, the reduced NAD, you can see that, becomes oxidized, and this hydrogen is given to make TP. One TP is recovered from one cycle. The other 5 TPs, look, they undergo another very series of biochemical pathway to recover another RUBP. 3 RUBPs recovered from 5 triose phosphates. There will be combination of different chemical reactions to recover that. It is a very long process. You will learn that in detail, maybe in your further stages. But plants finally develop the 3 RUBPs which were already there, and they will again start to take another 3 carbon dioxide, and the process continues. So, how many cycles are there now? There should be 3 carbon dioxide to give you 1 triose phosphate, another 3 to give you another 1 triose phosphate, triose phosphate plus triose phosphate reaction, they make 1 glucose. So there should be 6 Calvin cycles to make 1 glucose. Then the process continues in every thylakoid membrane. It's not happening at once. Every thylakoid membrane inside the stroma. Of course, you have to know this process takes place in the stroma. This is a very important part. In the stroma, I mean every process happens until they make billions of glucose molecules. Combine them, store it in the form of starch in their stem, in their roots, in their seeds, everywhere they got as a storage structure, even sometimes in the leaves. And they consume it for their metabolic process, and the remaining stored food will be eaten as a food for us. We eat them as a potato, we eat them as a tomato, we eat them as fruits, seeds, all the grains we eat, injera we eat, is the result of this process. I need you to know those stages then. What happens during light dependent and light independent stage? In the light independent stage, carbon dioxide combines with hydrogen to make glucose. So carbohydrate is made. 2. The ATP formed during light dependent stage is used in the process of light independent stage. NADP, which was reduced in the light dependent one, is now oxidized, and the hydrogen it carried from water is given to GP to become triose phosphate. And Rubisco, the most important enzyme here, is used to combine carbon dioxide and RUBP. So events happening in each stage must be remembered. All right? This is the whole stage of light dependent and light independent stage. Water splitted, light absorbed, NAD, P, and ATP are taken. Of course, don't forget this is reduced. There is hydrogen here, and NADP returns back. Oxygen is released, light dependent. 1 triose phosphate, 2 triose phosphate, finally 1 glucose. Triose phosphate, triose, 3 carbon, don't forget it. 3 carbon, 2 3 carbon molecules combine to make 1 glucose. That finally becomes starch. Okay. So this is a comparison between the two. I just want you to summarize them. Okay? Light reaction or light dependent. Only in the presence of light it can happen. Okay? photochemical stage or phase, photo, light is required, happens in the grana of the chloroplast, where the thylakoids are. Inside the thylakoids, there are photosynthetic pigments. Look the depths it goes. Grana, thylakoid, then it goes to inside thylakoid. There are many photosynthetic pigments forming PS1, PS2. That is not PlayStation. Okay. NADP utilizes the hydrogen ion and becomes NADPH, reduced. ATP and NAD are the end products. Of course, water split. Water molecule splits into hydrogen and oxygen. What do you call this? Photolysis. Okay? Where does it happen? PS2, photolysis happens. What happens in PS1? Reduced NAD. ATP. ATP is made here. Dark reaction, it happens whether there is light or not. If there is light, it happens, but it doesn't need light, okay? It's called a dark reaction or light independent. Light independent is better because it doesn't require any light, even though there is light. It is a biochemical phase. It involves enzymes to produce glucose, not the light. Of course, light is used in the first stage. Takes place in the stroma, in the fluid-filled part of the chloroplast. The hydrogen of the NADP brought from this is combined with carbon dioxide. You know, carbon dioxide, when combines with hydrogen, it becomes reduced to carbohydrate, especially glucose. Glucose is the end product. ATP is used. NADPH are used in the process. Okay? Carbon dioxide is utilized. There is no photolysis, obviously. All right. So let's go to the other topic of this lesson. How do plants use different pathways of photosynthesis? You know, plants do not follow the same kind of pathway to make food. Some plants follow a C3 pathway. We call it C3 photosynthesis. Some plants use a C4 pathway. The whole process that we discussed so far is a C3 pathway that happens in plants that are found commonly in temperate zones. But plants that are found in very hot places like tropics, plants such as grasses like sugar canes, corn, okay, those kind of plants, as they grow in a very highly crowded situation, they are usually growing together. So they usually suffer from a different kind of challenges to develop another pathway of evolution to make a different pathway of photosynthesis. Okay, in C3 pathway, it takes place in plants living in temperate zone, and they produce, it's called C3 because they just make a carbon 3 molecule, GP, glycerate phosphate, which eventually reduced to become triose phosphate. Then they make it. There is no problem in these plants. Just the topic we discussed is about C3 plants. But what happens in these plants is the main issue. Photorespiration. What does that mean? Respiration, you know it. Photo is light. You see, these plants sometimes suffer from this process, which might reduce the efficiency of photosynthesis by 25%. Which means if they have to make 100 kilos of glucose, food in a day, they might produce 75% if there is photorespiration. When does this happen? This happens when they suffer from loss of or lack of carbon dioxide in the atmosphere. There will be sometimes less carbon dioxide in the atmosphere that leads them to suffer from this problem. So this C3 pathway, they follow, as I have mentioned, the same pathway that we discussed. But in low carbon dioxide concentration, in condition, Rubisco, as I have mentioned, it is an enzyme called ribulose bisphosphate carboxylase, then oxygenase. It can react with oxygen if there is no carbon dioxide. This makes a different unwanted product in the process of photosynthesis called phosphoglycolate. And this phosphoglycolate is not required in the process of photosynthesis, and it reduces the process. Instead of producing more GP, they produce another unwanted waste called phosphoglycolate, which takes another very long pathway involving mitochondria, peroxisomes, and even other organelles to make or to return phosphoglycolate back to GP. So photorespiration is a disadvantage for such plants. Okay? When C3 plants do this, the concentration of carbon dioxide in the leaves fall, and this might cause them. How do they face this problem? When conditions become hot, like sometimes when the temperature increases, there will be a higher rate of transpiration. And the transpiration will cause these temperate plants to lose more water. They don't want it. So they usually tend to close their stomata. When they close their stomata, it's not only preventing water loss, it's also preventing carbon dioxide from getting into the chloroplast. This reduces photosynthesis. And inside the chloroplast, if the carbon dioxide level is low, Rubisco doesn't have option to use oxygen rather than carbon dioxide. This might finally lead to reduction of the rate of photosynthesis. So, this has caused some other plants to evolve into other pathway so that they can efficiently make light, food out of light. Okay? Rubisco undergo this process. For example, you see it combines with oxygen in case of C3. And then it makes GP and one other phosphoglycolate. Not wanted. And this is again converted to phospho-GP into such process. From chloroplast to peroxisome, from peroxisome, mitochondria. Then it undergoes such pathways, again they recover the GP. Why? This is a waste. So the plants rather chose, what if we use another way? And they evolved into a different pathway of photosynthesis, C4 pathway. These plants are somehow very efficient in reducing photorespiration, almost totally. These plants made some other different technique. I want you to see that to avoid the problem of photorespiration, they have actually made some transitions here. They make one of the processes in the palisade cell, then they take the next stage in the bundle sheath cells. Bundle sheath cells are usually used for transportation, but in this case they could be used as a kitchen to make carbohydrate. Yes. Okay? So it's not only happening in the palisade cells. It goes back down to... So how? How does that happen? Okay? The first compound, C4 pathway, obviously, they make the first compound C4, carbon 4 molecule. Okay? Then they finally change it back to the one it is required in the process. But these C4 plants usually are the plants that are found in tropics, like our country. Maize, crabgrass, other same plants like sugarcane, sorghum. These plants make a C4 pathway so that they can usually use a different mechanism to make that happen. Okay? As we have very few time to actually cover the whole lesson, I want you to go through this very important part of the C4 pathway. This is the mesophyll layer and this is the bundle sheath cells. Look what happens. In the mesophyll cell, the oxaloacetate molecule, you see that it undergoes such a cycle, and this 4 carbon molecule is made, malate, oxaloacetate, malate. Then this pyruvate comes into the bundle sheath cells, and the carbon dioxide is recovered from there. Then the Calvin cycle takes place in the bundle sheath cells, not in the palisade, to prevent oxygen from interfering in causing photorespiration. Okay? Then this Calvin cycle finally makes the sugar, then just vascular tissues transport the sugar to where it is required. It happens in bundle sheath cells. What happens? The light independent stage. I just want you to summarize the whole view. If we go into the details of every step, it might require hours. So I need you to make that C4 plants use or make different cells to make photosynthesis happen. C3s, they actually do it in the palisade cells of the leaf. And we also see the other kind of cells called CAM pathway. the other kind of pathway of photosynthesis. The C4 pathway we have already seen. Why C4 plants experience a low concentration of carbon dioxide? As I have mentioned earlier, they grow together, okay? They grow very close together. That makes a very high competition of carbon dioxide. For that reason, they have to compete to take in carbon dioxide and make the food, all right? The last one is CAM pathway. These are the plants usually found in very hot places like desert, okay? Desert plants, xerophytes, euphorbias, okay? You know, these plants usually suffer a very extreme temperature. Very huge loss of water is suffered during daytime. So they have to keep their stomata closed during day, and they keep it open at night. And this affected their pathway of photosynthesis, as they need light and they need food. But this problem has caused them to develop a pathway called CAM pathway. This is relatively dependent not on where the photosynthetic process takes place. It depends on time. So how do they manage it? In the extreme heat of desert, having open stomata is a problem. So at night, plants open their stomata to allow carbon dioxide. They take in at night. They store it in the form of C4 molecule called oxaloacetate. Don't forget. This enzyme, PEPCO, just like Rubisco, okay, is used in C4 and CAM plants to make the 4-carbon molecule oxaloacetate. Then they change it to malate and store it in the vacuoles, and they finally, okay, this process happens at night. What happens during daytime? Light dependent stage happens. They absorb light, they split water into hydrogen and oxygen, and that hydrogen kept in the NADP, and NADP waits. ATP is made during day, it waits until the light goes. Then when the night comes, or when the light intensity decreases, they switch to the light independent stage, okay? And during day, the light dependent stage happens. Then this malate in the vacuole is broken down to glycerate molecule, which I have mentioned, the GP and Calvin cycle. Then carbon dioxide is released back, just like in the C4 pathway, and they make glucose using the same Calvin cycle. But they have to store this carbon dioxide at night, okay? So I will have to finish this with this lesson. I want you just to compare these two plants, okay? Identify C3, C4 plants. C3, bundle sheath cells are not used. Only palisade cells are used for photosynthesis. In C4, the bundle sheath cells have chloroplast with no thylakoids. No thylakoid means low light dependent stage. It only is used for the light independent stage, okay? Enzymes, Rubisco is used, PEPCO is used there. Temperature at which they take place. C4s usually are tropic plants, temperate plants. The temperature, you know that they are different, and they have also optimum carbon dioxide concentration. Where is carbon dioxide fixed? Mesophyll cells, okay? Calvin cycle, mesophyll cell, bundle sheath cells. You should know this comparison because you are mostly asked about the differences between the two. Sometimes we see 3, C4, and CAM plants as well. So I need you to very important points. You don't need to go very detailed as you are prepared for national exam. Focus on very important points. Follow the learning competence or the objectives, okay? This is a comparison between C4 and CAM plant. Very good example of CAM plant, pineapples, sugarcane. How they use this carbon dioxide. Carbon dioxide is stored in mallets, then converted to pyruvates, then this carbon dioxide again goes to Calvin cycle. This happens in the bundle sheath cells. They make sugar, and they use that sugar, and they give the sugar for us. In sugarcane, you know that. And again, in plants, the light-dependent stage happens daytime. The light-independent happens at night. Plants fix this carbon dioxide. They follow the same pathway like these ones. And this carbon dioxide is taken in. Calvin cycle produces the sugar. But it happens at night. And this one happens at night. This one happens day, night, it doesn't matter. But the light-dependent stage, which absorbs light, must happen daytime. The stomata are closed, but the process is happening inside. Okay? I finalize my lesson with this. It's a lot of topic. Okay, students? I need you to go through the main points. Photosynthesis is the process that plants harness light and convert it to food or carbohydrate. Sometimes even they might not change it to carbohydrate. They can change triphosphate to lipids, to proteins, to every kind of molecule, to nucleotides. Okay, every biological molecule we have is made from those processes. And it happens in two stages. Light dependent first, light independent follows. Light dependent gives ATP that is made by photophosphorylation. It also provides hydrogen through the hydrogen taxi called NADP. It also breaks down water molecule into hydrogen and oxygen. And this hydrogen, which is taken by NADP again, goes through a very long process of electron transport chain until it combines with NADP. In the light-independent stage, I want you to know that carbohydrate is made by reducing carbon dioxide using hydrogen. The NADP took hydrogen and combines with carbon dioxide. In the light-independent stage, ATP that's formed is used up. Okay? And finally, triphosphate is made and glucose is made. Food is served. Identify C3, C4 plants. Why respiration happens in C3 plants and why C4 plants evolved and how they reduced the problem that comes after low carbon dioxide concentration as they grow in a very highly crowded, very close situation. Identify Rubisco, RUBP, and also PEPCO, the enzymes. Thank you for your time. Have a good day.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called Photosystem 1 and Photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.